Hello, my name is Joshua Mugabe. I welcome you to Cytoskeleton and Cell Movement. What is cytoskeleton? What are its main constituents in animal cells? Cytoskeleton is the cytoplasmic structure that supports the cell. It keeps its shape, fixates, and moves the cell organelles. It is made of extensive network of fibers dispersed in the cytoplasm, anchored in the plasma membrane. Its components are microtubules, microfilaments, and intermediate filaments. Of which substance are microtubules made? In which structures and cellular processes do microtubules participate? Microtubules are made of consecutive dimers of the protein tubulin. Each dimer has an alpha and a beta tubulin associated. Microtubules participate in cell division. They are constituents of cilia and flagella. They also form the centrioles. Of which substance are microfilaments made? What are the properties of these elements that give motility to cells? Microfilaments are made of actin, a protein. The contractile association of actin with myosin and other cytoplasmic protein give the microfilaments the ability to promote cell movement. What are cell movements? How are these movements created? Cell movements are movements performed by the cell structures, like the movement of the cilia and the flagella, the pseudopody movement in amoeba, macrophages, and many others. The psychrosis of the cytoplasm and the sarcomere contraction in the muscle cells. Cell movement can be created by the cytoskeleton action, by differences in the viscosity among the cytoplasmic region, or by the intracellular contraction systems. What are cilia and flagella? How do these structures acquire movement? What are some examples of ciliated and flagellated cells in humans? Cilia and flagella are structures found in some prokaryotes as well as in some eukaryotic cells. They play defense nutrition and movement roles in the cell. Eukaryote cells of protists and animals, they originate from the centrioles that migrate toward the plasma membrane and differentiate into the structure projected outside the cell. Each cilium or flagellum is made up of 9 peripheral pairs of microtubules and 1 central pair, all covered by membrane. In bacteria, flagella are made of a protein named flagellin, and there can also be fibria made of pilin. The fixation base of each cilium or flagellum in the plasma membrane, they are proteins that work as muscular motors, providing movement for these structures with energy spending. Due to this energy spending, ciliated and flagellated eukaryotic cells have a large number of mitochondria. In human, ciliated cells can be found, for example, in the bronchial, tracheal epithelium. These tissues, the cilia, have a defense function for sweeping the mucus and foreign substances that enter the airways. Sperm cells are a typical example of flagellated cells. Their flagella is the propulsion equipment for the movement towards the ovule. How does the amoeboid movement occur? What example of beings and cells that use such movement for locomotion? Amoeboid movement are created by cytoplasmic movement and plasma membrane projection called pseudopodia. Their formation actively changes the external shape of the portion of the cell surface, making it move along the substratum. Pseudopods appear from differences of viscosity among neighboring regions of the cytoplasm near the plasma membrane, and from the contractile action of microfilaments. Amoeboid movement occur, for example, in amoeba, protozoa, organisms that use their movement to find food. The leukocyte cells of the immune system, when attracted by chemical substances, immune mediator, use amoeboid movement to get out of the capillaries in region of tissue damage to participate in the inflammatory process. What are some examples of movement created by contraction of sarcomere of the muscle cells? Handling a cup of coffee, peristaltic movement of the bowel, cardiac beat, and even a smile are all examples of movement created by the contraction of the sarcomere of the muscle cells. These contractions is a type of cell movement. What is cyclosis? Cyclosis is a type of internal cell movement in which an oriented flow of circulating material is created and maintained in the cytoplasm by the actin of microfilaments. Cyclosis is more easily observed in plant cells. Thank you very much for participating in cytoskeleton and cell movement. Please do like, share, and subscribe.